Cell culture is the first step in the biopharmaceutical manufacturing process. It involves growing cells in a laboratory environment to ensure there are enough cells to make a biologic. Cells grow by splitting in half, changing one cell into two cells. As this happens over and over again, the cells grow exponentially until there are enough cells to make it into this step 2 bioreactor. The cells need to be grown under controlled conditions, ensuring that the critical quality attributes are met. Some of these critical quality attributes include protein concentration and purity. Scientists would measure the protein concentration and purity of the cells to ensure that they are ready for the bioreactor. The way that they can ensure that the protein concentration and purity are going to be correct is by ensuring that the critical process parameters, or CPPs, are set properly. Some of these CPPs could be nutrient amounts, carbon dioxide and oxygen concentration, pH, temperature, and other factors depending on what type of drug they're intending on manufacturing. These CPPs would be measured throughout the cell culture process and would help scientists ensure that the critical quality attributes would be met at the end of the cell culture step. Cell culture is also subject to regulation, as any contamination in the cell culture could threaten the drug or the viability of the drug. To do this, cells have to be cultured in a sterile environment where there's no chance of anything else getting into the culture besides the intended nutrients and other additives, as well as the cells. Guidelines laid out in the ICH-Q5A guideline help biologic manufacturers comply with the regulations, ensuring that their laboratories are sterile and therefore they're meeting all the regulations laid out by regulatory bodies.